I do want to thank you for having me. I mean, this is really wonderful. I'm thrilled to see this concerted effort to bring information about clinical research to Argentina and throughout South and Central America and other Spanish-speaking countries. I think, one of the things that I really feel convinced of is that this kind of concerted effort raises awareness, and also I think lets companies and investigators know that there's a population of people who would really like to be a part of clinical trials, and I think allows us the potential to expand. And that's really, really exciting, I think. I'm going to talk today about a program called RAPA-501. And RAPA-501 is not a drug or a medication as we think about it. It actually is a cell therapy. And these are reprogrammed T stem cell therapies that we're bringing to ALS, and all that will, I think, make a little more sense as I go through the presentation. This is a big collaborative effort. I'm one of the principal investigators for the project. I work alongside colleagues Suma Babu and Sabrina Paganoni. We're working in very close concert with the wonderful team at RAPA Therapeutics. They are the ones who hold the investigational drug approval for this to go into trials. And this is generously funded by the National Institute of Health, specifically the NINDS, or National Institute of Neurological Disorders and Stroke. And I'll talk a little bit about some of the funding that came from there. Now, the program that I'll talk about today with RAPA Therapeutics is really built on science from 2 trials that showed the promise of treatment with what are called regulatory T cells for ALS. Now, this is science that was done by a wonderful physician and researcher at Methodist University in Houston, Texas, showing that treating people who have ALS with regulatory T cells, which are T cells that quiet the immune system, has the promise of slowing ALS. And RAPA Therapeutics saw that and sort of wanted to build on that really wonderful, exciting result. And it built into the RAPA-501 program. I want to start with just a little terminology. T cells are a kind of immune cell. So our body has lots and lots of different immune cells in it. But some really central ones are called T cells. And T cells are responsible mostly for increasing inflammation. Now, there are many types of T cells. If you open an immunology book, they'll talk about many, many different kinds. I have 3 kinds written here on the slide, and 2 of these are going to be most important for today's talk. But the 3 I've written down are one group called killer T cells. Those really fight off cells that have in the body that have already been infected. Helper T cells. The helper T cells help the killer T cells do their job. So it's really a way of fighting infection. And then the last kind of T cell that I'll talk about, and this is where I'm really going to focus most of my talk today, is on regulatory T cells. These are an unusual kind of a T cell. Regulatory T cells actually reduce the immune response. So if you think about, if we become infected, let's say you get an infection, your immune system responds, ramp up your immune system, and then the infection is fighted off, your body fights off the infection. And then after that, the immune system quiets down again. And the regulatory T cells are the ones who are responsible for quieting that immune system down. So they have anti-inflammatory characteristics. So again, the 2 that I'll talk about most today are the helper T cells, which are pro-inflammatory, and the regulatory T cells, which are anti-inflammatory. And one of the problems in ALS is that regulatory T cells seem to be reduced so that Th1 and Th2 cells, these subtypes of helper T cells, are overactive. Okay, so that's a finding that has been replicated in many labs. There are too few regulatory T cells, and the helper T cells are overactive. RAPA Therapeutics has actually a long history of cell therapy, specifically looking at T cells. The founders of the company were at the NIH for nearly 2 decades, learning about T cells, learning about killer T cells, helper T cells, regulatory T cells. And they formed RAPA Therapeutics actually initially to work on cancer. And in cancer, what they wanted to do and what they are doing is increasing killer T cells and helper T cells to increase the immune response to cancer and help the body fight off cancer. But they also saw the opportunity to test out regulatory-like T cells, Treg-like cells, as a way of fighting ALS. So in 2018, they treated a single person with RAPA-501 cells, and then moved on to a phase 1 trial that I'll tell you about, where we treated a series of people with this, and then designed a phase 2/3 trial, and we've been working on the expanded access protocol alongside all of this. So the program really has made a lot of progress in the last 5 or 6 years. Now, the central premise here is that neurodegeneration leads to inflammation, and that inflammation increases the rate of neurodegeneration. So here you can see a motor neuron, and this motor neuron, let's imagine, has begun to become ill with ALS. As that happens, there are a number of molecules called cytokines and chemokines that attract immune cells. So it sends off all these molecules. The molecules encounter helper T cells, Th1 helper T cells, and they call them to this area where the neuron is injured. Th1 cells come in, and they assume that this is an infected cell, and they start to attack it. And they might also attack surrounding cells, and that begins to hasten the process of neurodegeneration. Now, our body has a natural way of reducing this response. So naturally, at the same time that helper T cells are activated, regulatory T cells, or Tregs, are also activated. And these Tregs actually quiet the response of the helper T cells and try to keep this from getting out of control. So there's a balance that our bodies naturally have. The problem is that, as I said before, in ALS, these regulatory T cells are reduced in ALS. So we have fewer of them, they're not working right. And what happens is we get more inflammation, and these now pathogenic, now problematic helper T cells really continue their attack on the motor neurons. And what we want to do is circumvent that attack. And one of the ways that we might be able to do that is through RAPA-501 cells. So I've told you now that there are a number of different kinds of T cells. Most of them are pro-inflammatory. There were 2 that were important today, which are the helper T cells and the regulatory T cells. These Tregs are reduced. And now RAPA-501 cells can be added to the mix to supplement the Treg activity. So these RAPA-501 cells are going to come in and take over the role of regulatory T cells and reduce inflammation. And I'll tell you about how that happens in just a moment. But their main goal is really to combat this helper T cell inflammation. So here we can replace what had been these regulatory T cells in our diagram. We can replace this with the RAPA-501 cells. Those RAPA-501 cells will inhibit the inflammation and then stop this killing of the motor neurons. That's how we envision this happening, and that's why we're excited about this program. That can allow these motor neurons more of a chance to sort of regulate themselves and hopefully slow the spread of the disease. So RAPA-501 are what are called autologous cells. That means that they originate from the patient, him or herself. And the way that we sort of make these RAPA-501 cells is that T cells are actually harvested from the participant, from the person in the trial, through a process called apheresis. This is like a big IV that goes, takes the blood out of the body to a centrifuge where the cells are sorted, and only the white blood cells, only the immune cells are kept, including the T cells. All the rest of that blood goes back into the veins to the other arm. And those cells are then shipped down to the manufacturing facility at RAPA Therapeutics. And step 2 is those T cells are de-differentiated. So we have all kinds of T cells. There are killer T cells, helper T cells, regulatory T cells, many, many different kinds of T cells. And they're all through a process that doesn't involve manipulating genes or anything. It's just how they're grown. They're reverted back to a very naive state where they can become any kind of T cell. And then they're redifferentiated. So those cells are then taken from a very naive state into this special RAPA-501 state where they look a lot like a regulatory T cell, but sort of like a helper T cell as well. And I'll tell you about why that's a benefit in just a minute. They don't look just like regulatory T cells, but they've got new characteristics as well. After those cells are created, then they're infused back into the patient, into the trial participant, to be anti-inflammatory. And so the cells themselves are like a living drug. Now, I told you that these cells have characteristics that are like a regulatory T cell and some that are like a helper T cell. And that's important. There are a number of things that happen to these cells when they're redifferentiated. There's a process called mTORC1 and 2 blockade that really stops these cells from turning into other kinds of cells. So sometimes when you put regulatory T cells back into somebody's body, they can actually switch into killer T cells or helper T cells, which is exactly the wrong thing for them to do. So that through this process that RAPA has developed, that possibility is taken away, and these cells will stay the cells that they are when they're put into the body. They have a process for selecting for the most metabolically fit cells. It's sort of survival of the fittest when they grow these cells, and they really select for ones that are fit. And they also sort of remove ways that the body could manipulate these T cells into doing causing inflammation in the body. Importantly, the cells retain markers on their surface that allow them to home in on inflammation. In other words, we put them into the bloodstream, but they can actually find where chronic inflammation is going on at the level of the motor neurons. And that's a really important part of these cells, so that they find their way to the motor neurons. Now, this is just a sort of fancy immunology way of showing that these RAPA-501 cells express markers that make them both like a regulatory T cell, quiets inflammation, but also like a helper T cell. And you say, well, isn't it bad that they're a helper T cell as well? The answer is, when they're a combination of a regulatory T cell and a helper T cell in this way, they find their way to the inflammation better, they stay the kind of cell that they are better, they're more resilient, and they're actually more effective as regulatory T cells than sort of regular, just sort of putting back in regulatory T cells, based on the science that RAPA has done in the lab. And so the science that they've done happens in a Petri dish. And in the Petri dish, what they find is that these RAPA-501 cells, if you put them in the same Petri dish with inflammatory cells, cells that are from the brain and the spinal cord called microglia, the RAPA-501 cells suppress the inflammation. So you'll see in orange in these figures over here, you'll see that in orange, this is an untreated state, and in blue, this is the state where we have the inflammatory cells and RAPA cells. And you can see that there are lower levels of IL-6, IP-10, interferon gamma. So all of these are pro-inflammatory molecules, and the RAPA T cells have reduced the amount of those molecules. So there's less inflammation in this Petri dish, in this experimental environment. So that's really exciting. What it means is that these RAPA-501 cells are actually suppressing inflammation. And that set the stage for us to go to a phase 1/2 trial. And I should say there was also a lot of characterization of these cells, a lot of safety of these cells to make sure that they're not infected. So there's a huge amount of work that goes into this. I'm just showing one, I think, salient slide here. So then we started with the RAPA-501 phase 1/2 trial. And there were 2 centers that were enrolling participants, Mass General Hospital and Hackensack Hospital in New Jersey. And we enrolled 3 groups or cohorts of patients, and each group had between 3 and 6 participants. We started with a group that got a low dose of cells, that's 20 million cells per injection, per infusion. And then we moved, the plan was if that looked okay, which it did, then we would move to a second group or second cohort of participants who got a higher dose, up to 80 million cells. And if that looked okay, we would then test a protocol where we treated people with the high dose of cells, 80 million cells. And before we gave them the cells, we would give them a little chemotherapy to try to what we call make room for the new cells. So try to reduce the number of T cells that are in the person's body already that may not be doing their job right, and put in the 501 cells. The chemotherapy that we used were two common chemotherapeutic agents called cyclophosphamide and pentostatin. These we gave them at very low doses. They're really targeted specifically at lymphocytes, and we did actually see the T cells reduce a little bit. And the idea was to see whether there was more effect with that preconditioning chemotherapy. The phase 1 trial and these 3 cohorts or groups of people that we treated had really 3 major aims. We're always doing a lot of things at once, but these were the 3 major aims. First was evaluate the safety and tolerability. Were there going to be side effects we hadn't thought of or hadn't seen in some of the previous experiments? Were people able to tolerate both the low and the high dose? What kind of side effects would we see? The second thing was to evaluate the effect of these RAPA-501 cells on inflammation, as well as on neurodegeneration. And we did that by taking blood. We took it very frequently, and we looked at inflammatory markers, and we looked at markers of neurodegeneration. The most common one that we know of is called neurofilament, which is a scaffolding protein. It's like the protein that helps make up the structure of the motor neuron, and that goes up in ALS. And then the third thing we evaluated was any effect of RAPA-501 on clinical measures. And that means how long people live, what their function was like, how they're breathing, how strong their breathing, muscles of breathing were. And this was really exploratory because with a very small number of participants like this, it's hard to make many conclusions, but always worth looking at. There were a lot of visits, so each one of these circles represents a visit in this phase 1 trial. The first is what we call a screening visit, where people come in and find out whether they can participate in the trial and whether that trial is one that they'd like to be a part of. We draw blood, we get clinical information, we do exams. And if people passed that screening and were eligible for the trial, then they went on to have this apheresis procedure where the T cells were harvested and sent down to RAPA Therapeutics. It took about 4 to 6 weeks to manufacture the RAPA-501 cells, and then the first infusion visit happened. And that infusion would last just about 15 minutes. The visit was longer than that, but it's a short infusion. After that, on day 1, day 14, and day 28, participants would come back to our center and we would draw blood so that we could look for biomarkers. And then the subsequent cycles were really very much the same. So the second infusion would happen 6 weeks after the first one. And then people would come back in day 1, day 14, day 28, and 2 weeks later come in for their infusion. We did this up to 4 times. Now, we were still working out manufacturing. Not everybody had enough cells that we could do all 4 infusions, but they would come for as many of the 4 infusions as possible. And we would follow them all through that time. And then we actually followed them long term so that after they completed this final infusion, we would see them 1 month, 3 months, and 6 months afterwards to see whether what we did to their immune system had lasting effects. It's possible that the benefit would be long lasting. And I will tell you right now, this is a lot of data for us to analyze. And in fact, that analysis is still very much going on. We do have a little bit of data to say that we saw a very strong reduction in pro-inflammatory levels. So this MDC is sort of a pro-inflammatory level. So this is the pre-therapy level, and we see a reduction of up to 99%, 1 day, 8 days, 28 days, 42 days, 50 days, and 70 days after the first infusion. And so these are all these biomarker visits that we had people come in for, and then the second infusion, and then the biomarker visits after that. So we're seeing a very strong effect on inflammation. And then we sort of looked by cohort. So this was sort of, the participant number. These first two participants had the low dose. Then we had a couple with the high dose, and then we had a couple with the high dose and the chemotherapy. And what we really see is that there's between an 88 and a 99% reduction in inflammation in all of these participants, except maybe the last one. The last one or two were getting the chemotherapy as well. So if anything, the chemotherapy certainly didn't seem to be adding anything. It also gave us some confidence that the low dose was maybe enough, but the high dose was well tolerated. And so, I think, going, somewhere between the low and the high dose, either using the high dose or the low dose should be sufficient to target immune function. So we learned really a lot already, and we have a lot more to look at from that trial. Based on this, there is a design for a phase 2/3 trial. In fact, it's been approved by the FDA. This is designed as a placebo-controlled, randomized controlled trial, again, with 4 treatments, 6 weeks apart. There are no blood biomarker draws between visits to make that more simple. And the outcomes really focus on clinical outcomes like ALS functional rating scale and survival, vital capacity, safety, and then some more exploration of the immune and neurodegeneration biomarkers. This is a new area, and so I think biomarkers will continue to be important. Enrollment has not started for this program. In the meantime, we did have the opportunity to start an expanded access program of RAPA-501. And this was funded through an NIH mechanism that was specifically meant to fund expanded access programs of novel therapeutics for people with ALS. And we were excited to work with RAPA Therapeutics to get this project going. The NIH Expanded Access Program in ALS is a grant program for research that uses data from expanded access to investigational drugs. And what that means, what I mean by expanded access is it's a program designed specifically for people who are not otherwise eligible for ALS-related clinical trials. It may be somebody whose disease has progressed too long or who has had the disease too long for them to be eligible for other clinical trials. So it's a new population. It's one that we don't see in clinical trials, people with a little more advanced disease. But it's also giving them a chance to have access to experimental therapies and the hope that comes with that. This is funded specifically by a bill that was passed through Congress with really a lot of support from the ALS community, and particularly people living with the disease. There's one stipulation that it can't interfere with the ongoing clinical development. So we have to enroll people who aren't eligible for the clinical trials that are going on. In this case, what we did was define that people had to have a vital capacity, that is a lung volume, less than 50% predicted for their normal. That means people who have, breathing that's been substantially impaired by ALS. And this typically means that they won't be eligible for other ALS trials. But otherwise, we left the inclusion criteria quite open. Sporadic or familial disease. They can have really any category of LS-Gorriello criteria, which is a way of sort of categorizing ALS. They can be on other medications. There's no restriction on time from diagnosis. There is one characteristic that is true, which is they have to have a certain number of T cells. If they don't have enough T cells at the outset, and they can, we don't differentiate between killer T cells, helper T cells, regulatory T cells, but if they don't have enough T cells, then we won't have enough cells to make the product or the RAPA-501 and give it back to them. There are two main aims of this program. The first aim is to provide RAPA-501 to a different population of people living with ALS. There are 40 participants who will be involved in this program. Again, they wouldn't be able to access this through other trials because there are cutoffs for vital capacity and how long people have had the disease, so that we can study this in a group of people who are early in ALS and may be most likely to respond. And we also want to ensure that we're recruiting a diverse population of people living with the disease. And then the second aim is to to really leverage this opportunity to collect rigorous scientific data to evaluate the effect of RAPA-501. So safety in people who have more advanced disease or more rapidly progressive disease, tolerability. And we're going to collect a lot of blood for biomarkers. I'll show you how we'll simplify this, but we'll still have a lot of opportunity to look at biomarkers. Now, inflammation changes over the course of ALS. We know this from previous studies that inflammation may be more significant later in the disease. And so we want to know, do these RAPA-501 cells still stand a chance of reducing inflammation even in people later in the disease? So it's a really important opportunity. We'll look at neurofilament, which is a biomarker of neurodegeneration, and then clinical outcome measures, including ALS functional rating scale, a new scale called the Rhodes, breathing capacity, vital capacity. And we're also leveraging some new technology with a group called Everything ALS, which is an ALS nonprofit group, to do remote monitoring, where people actually have sessions from their home. And we collect surveys, the ALS-FRSR and the Rhodes. We collect pulmonary function using a vital capacity. We collect speech samples because we can see that speech changes over the course of ALS. And we're even using accelerometers, that is sort of wrist-worn accelerometers, to see how people are moving. And so we'll have a huge amount of data there as well. We tried to make this as rich a sort of data source as possible. Again, there'll be 40 participants. These will be people participating across 10 trial centers. We talked about the primary and secondary objectives, and even the exploratory objective of getting data from people's homes. These are the 10 centers that we have lined up. Many of these have been underway, starting up, and are beginning to enroll participants over the next month or so. I told you that we would look at how we've simplified the visit schedule. And again, each of the circles is a visit. So people come in and they're interested, they have a screening visit. 40 will be, we'll be able to screen up to 40. They'll have their apheresis. We'll begin doing our at-home surveys. So these at-home surveys, speech collection, accelerometry, and breathing capacity will all be measured throughout the study. And then the only visits that really happen are the infusion visits, 1, 2, 3, 4, 6 weeks apart. And then we have 2 remote follow-up visits at the end where the person is just seen over a video conference like this. So we're really excited about the program, about the phase 1 and analyzing the rest of the data that we have about the expanded access program and about the phase 2 and 3. I want to thank all of the clinical trial participants and their families. It is really an incredible thing for people to take the time and really put forward all the effort to be a part of clinical trials. And we couldn't make scientific progress without people who are willing to be participants and their families and caregivers and loved ones and friends who support them. The NIH funds this. My entire team at Mass General and my colleagues, Serena Paganoni and Suma Babu, the team at RAPA Therapeutics, Dan Fowler, Jenny Sunga, Sylvia Yip, and many others, just an incredible team to work with. The clinical research organization that we're working with to help this trial go is called Osmosis. There are 10 clinical trial sites with investigators and coordinators and nurses who are working diligently to get this going, and we really appreciate them. We'll be working with statistics from Origin, data sciences. Everything ALS is providing all of the at-home monitoring. The ALS Association and ALS Northwest have really helped to get the word out about this trial as well. Thank you very much.
Excellent. Thank you so much, Dr. Berry. That was a very impressive presentation, and it looks like really promising, really hopeful for those ALS patients and their families that are going to be watching this conference. And so thank you very much for that. I just have a couple of questions for you. So it looks like the technique and the procedure, let's say, of this clinical trial of RAPA-501, at least from the non-scientist perspective, at least from the perspective of someone who is a family member of a person who had ALS, it looks pretty innovative. So what would be the new element that this kind of clinical trial is bringing into the field right now?
Yeah. Yeah, no, I think it's really exciting, and it is also fairly new. I think cell therapies in general are a relatively new area in science, and we see more and more cell therapies in neurodegeneration as well as in other diseases like multiple sclerosis and in fact some diseases outside of neurology as well. I think one of the things that's really promising about cell therapies is that they may have multiple effects at the same time. We know that cells don't do just one thing, but they do many, many things. And so I think one thing that's really innovative here is we're not hitting a single target, but we're hitting a sort of a broad set of targets, and we're doing that with a living cell. That also creates some of the challenges, and I think the reason that we don't see many of these cell therapy trials all at the same time is that they're expensive to create the cells. And the science behind it is complex, and we need to really be sure that we're having the effect that we want to have, that these cells stay the kind of cells that they are when we put them in. So if we put in a regulatory T cell, we want it to stay as a regulatory T cell. We don't want the body to change it into a different kind of cell. And so that's been a lot of the innovation that goes into this particular project.
Great, thank you. So what's next? What are the next steps? We know that from your presentation, you already explained that this trial is just getting into a phase 2-3 environment. So what are the next steps in terms of the of publication of results? And this is a difficult question, but I have to ask. What is the potential for conditional approval in the next future?
Yeah, so let me take this one step at a time because I think there's a lot of work in front of us. First, I think there's much more data to get from the biomarkers in the phase 1/2 trial, and those analyses are underway, and we have every intention of analyzing that data when it comes back and then putting it into a publication so that we can really say all the amazing things that we learned in that phase 1 trial. The expanded access program is underway, and so there are, as I said, 10 sites. One is open now. There are a handful of others that will be open before the end of next month. And then I think the remaining ones, hopefully in the month after that. So we have a lot of work going on at a lot of sites to get up and running with the expanded access program and get 40 participants enrolled as quickly as we can, because we know that people are waiting. So those timelines really matter a lot, and we take them very seriously. And we're very excited to make that progress. And then we'll have a lot more data to analyze. In addition to the biomarker data and the clinical data, we'll have all of the digital data from everything ALS, accelerometry, speech data, patient-reported outcome measures. So there'll be a lot of additional new kinds of data that we can analyze. And I think that'll be really exciting. And then the phase 2/3 trial, we want to get off the ground and running as well. And we're actually in the process of writing some grants for what may be more than one phase 2/3 trial, depending on how the program comes together. So I think 3 different fronts, a lot of work going on at the same time, really dedicated teams, and a lot of fun to work together and to see the progress that we're making. I wish we could make this all even faster, but we're working as fast as we can. I think I'm always hopeful when we go into a clinical development program like this or others that we will find positive results. And I think as far as approval, I think there are some important things to know, which is that we really need to focus on showing a benefit. And once we've shown a benefit, and if we've done those studies in a rigorous way with good methodology, then then the plan is you talk to the FDA about about the data that you have in hand, and whether that's enough to think about approval, or whether there need to be more studies to really continue to investigate. So, I think at this stage, the really important work is make sure the methodology is right, make sure the trials are designed well, make sure that we can move through those trials as quickly as possible, do good analysis, and that will hopefully guide us to exciting results.
Makes sense. Great. Thank you. So speaking about the current landscape in ALS research, and also speaking about conditional approvals too, we did have a couple of setbacks lately in ALS research. So what is your take on that?
Yeah, we did. I guess what I would say is I think that the process is working. I think we did see that there was some regulatory flexibility from a number of the regulatory agencies to work with the best data that we had in hand, and that we continued to get really important data that unfortunately didn't in the case of the Amylyx program, did not show a benefit in the second phase 3 trial. And that was, that was hard. But I think the system worked well, and we got all the data that we needed. They're continuing actually to gather more data, more long-term survival data from that program. So I think these pieces did what they needed to do. What's disappointing is that we don't have a drug that looked beneficial in all the trials, but we move along, and I think there's more science now than there was previously. If you look back to the beginning of that development program from Amylyx, there were a lot of things we didn't know about the biology of the disease that we do know now. So, for example, neurofilament is a much more established biomarker now and can guide our trials. We still have questions about it. There's still more to be learned, but we've learned a lot in the last 5 years or so. And we're using it very differently in trial design. We're also beginning to explore markers of TDP-43 function. So TDP-43 is a molecule that's central to the problems in ALS. It becomes sticky and gummed together and doesn't do its job of helping the cell make other proteins. And we can see now that there are other proteins that are made wrong, and they may be drug targets or biomarkers to help us understand whether we're changing the biology of the disease in important ways. So our tools for doing these trials are really getting better faster, and our drug targets are expanding. So, yes, there are setbacks. I think that's a part of doing hard science. That's why we do this. That's why we are dedicated to it and not just dabbling in it. But we keep moving forward because there are green shoots and there are really exciting, developments.
Yes, I agree. And moving on, how international is this RAPA-501 clinical trial becoming in the next future? I mean, we have a lot of folks from Argentina and other countries that are really eager to participate in these clinical trials. You already explained that there is an expanding access program stage attached to this clinical trial, so that's interesting too. Can you expand a little bit more on that?
Yeah, so I think the trials that have been done, and I think the ones that are being planned probably are primarily US based. It doesn't mean that people can't come to the US to participate in them. Because of the nature of the trial, the infusions have to happen at a clinical center. But I also think that there are huge opportunities for doing this program, but other programs doing ALS trials internationally. And for a disease that's uncommon like ALS, I think we need to really begin to make the case to ourselves as researchers, to companies that are often designing, the large trials, to regulators, that it's really important that we work together. We can do these trials much faster if we have, access to people that want to be a part of trials around the globe rather than in just a small handful of countries. And, I would say that, having patient advocacy organizations and having researchers and having active, people with lived experience in countries really does elevate the science, elevate the chances of having clinical trials, and elevate the quality of clinical trials in the country. So really a big hats off to you and your organization for really sort of raising the bar and raising the possibility of doing trials in Argentina.
And yes, we are pushing for that. Okay, so last question. Can you tell us about any other research project that you are involved with right now besides RAPA-501? I know you are involved in many of them, but can you maybe mention one or two of them?
Sure. Yeah. I want to talk about one that's not a clinical trial, but actually a clinical research project that goes by the name All ALS. And we're just enrolling the first participants in All ALS. And this is a program that I think really is, in some ways, the antidote for the kinds of setbacks that we talked about earlier, which is it's aimed at gathering clinical information, blood, in some cases, spinal fluid, digital data, and really bringing that all together and housing it in one place, all connected to each other. There'll be genomics as a part of this as well, and probably broad biomarker studies. All of that data, once we gather it, will go into a central data repository, which will be shared internationally with researchers who are trying to understand ALS better. And again, this is where I think our leaps forward in understanding the disease in people can come from. So, even if we look at things like neurofilament, a lot of the advances we made in understanding neurofilament as a biomarker came from existing samples and existing clinical data. And now what we want to do is bring even more kinds of data together and do that in thousands of people with ALS, and then make that available broadly. So that's, I think, a really exciting effort to help supercharge our information about the disease and finding new targets.
Excellent. Thank you. That's good news. Okay, Dr. Berry, once again, thank you so much for sharing some part of your very busy schedule with us for the ALS community in Argentina and other Spanish-speaking countries. We are looking forward to hearing more news about RAPA-501, of course, and your other research projects. And we are also looking forward to seeing you in Montreal in December.
Absolutely. I'm excited to see you there and excited to see others there. I can't say it enough times. I think really excited about what you're doing, in Argentina. And I think it will elevate the science and the care and the availability of trials there. And I'd love to be a part of that. So thank you for having me here, and I look forward to working together.
Thank you so much, Dr. Berry. You have a great day. Thank you. And we will be in touch, of course, as always. Thank you so much.
Bye-bye.
Bye.